Welcome back to The Explainer. Today we're doing something a little different. We are absolutely not just doing some dry, boring biology review. Oh no, we're actually going on an exclusive VIP tour of a bustling, highly advanced microscopic metropolis. We're going to shrink right down and explore the fundamental unit of life itself, the cell. I'm talking about a place with its own power plants, security borders, data centers, and manufacturing hubs. All right, let's dive right into this incredible world.
Here's the roadmap for our tour today. 1. Discovery and basic unit. 2. Plant versus animal cells. 3. Cell walls and membranes. 4. The nucleus, our control center. 5. Organelles, the cellular workforce. And finally, 6. Osmosis and diffusion. Let's get going.
Kicking things off with Section 1, Discovery and Basic Unit.
So, what do a brick wall and a human being actually have in common? I know, it sounds like a totally random question, but the answer is exactly what makes biology so fascinating. You see, both of them are built from tiny, independent structural units. Just like you literally can't have a brick wall without individual bricks, you simply can't have a living organism without cells.
And that brings us to this amazing piece of history. These hand-drawn notes detail Robert Hooke's massive discovery back in 1665. So Hooke was looking at a super thin slice of cork, which is basically dead plant tissue, under his own kind of primitive compound microscope. And what he saw was this honeycomb structure made up of all these little empty boxes. Because these tiny compartments reminded him of the small, sparse rooms that monks lived in, he actually coined the term cell, which comes from the Latin word cella, meaning small room. This was literally the very first time anyone had ever described a fundamental biological structure.
Moving right along to Section 2, Plant versus Animal Cells.
Now, a really crucial thing to understand here is that not all cells are built the exact same way. As this breakdown shows, we're essentially looking at two totally different styles of cellular architecture. Plant cells, they're like these rigid fortresses. They have a fixed rectangular shape thanks to a really sturdy cell wall made of cellulose, plus a massive central vacuole they use for storage. Animal cells, on the other hand, are more like flexible, dynamic, sprawling metropolises. They completely lack that rigid cell wall entirely, which gives them an irregular, variable shape. And their vacuoles, they're just small, temporary little storage units.
And here's the visual proof of exactly what we're talking about. In the plant cell, you'll immediately notice that incredibly thick outer wall acting as a rigid boundary. Plus, there's that huge central vacuole just dominating the interior space. It's so big it actually pushes the nucleus off to the side. Contrast that with the animal cell, which is bounded only by a thin, flexible cell membrane, and its nucleus, it sits proudly right in the dead center, kind of like a downtown city hall.
All right, Section 3, Cell Walls and Membranes. Let's talk about securing the borders.
You know, every great city needs a secure border, right? Well, the cell uses something called a selectively permeable membrane. And guys, this isn't just some dumb, solid wall. It's a dynamic, highly intelligent security gatekeeper. It completely controls the movement of substances, so it allows the essential things like oxygen and water to pass through freely while strictly restricting large molecules and keeping out any dangerous intruders. To break this down a bit more, the cell membrane basically acts as both a gatekeeper and a protector, carefully managing both passive and active transport. But remember how we said plant cells have that extra layer of defense? That rigid cell wall provides serious structural support. But here's the really cool part. It also creates what we call turgor pressure. Basically, when water rushes into a plant cell, the cell wall physically prevents the whole thing from popping like a water balloon. It keeps the plant upright and crisp instead of wilted. You know that drooping, sad houseplant you forgot to water last week? Yep, that is literally a loss of turgor pressure. Up close, this highly intelligent boundary operates on what's known as the fluid mosaic model. Just check out the lipid bilayer here. It's got these water-loving, hydrophilic heads on the outside and water-fearing, hydrophobic tails tucked away on the inside. It's totally fluid and dynamic. And those big, colorful structures embedded throughout? Those are integral proteins. You can think of them as dedicated tunnels and active transport pumps, constantly pushing molecules against the gradient to keep the city's resources perfectly balanced.
This brings us to Section 4, the nucleus, the control center.
Seriously, just look at the sheer level of security surrounding the nucleus here. It's got this double membrane nuclear envelope that is covered in tiny little pores, all designed to strictly control exactly what goes in and out. Inside this jelly-like nucleoplasm sits the chromatin network. This is essentially a tangled mass of DNA, the actual hereditary instructions for quite literally everything the cell does. And right in the very middle of it all is the super-dense nucleolus, which is where ribosomes are synthesized.
This whole structure coordinates absolutely all cell activities. It truly is the brain of the cell, or, the central control tower.
Okay, moving on to Section 5, Organelles, the cellular workforce.
So let's dive into the actual factories that keep this whole microscopic city running. Think of them as a highly sophisticated supply chain. First up, you've got the endoplasmic reticulum, or ER for short, which is busy synthesizing proteins and lipids. It hands those products off to the Golgi apparatus, which basically acts as the cell's post office, carefully packaging and secreting these products. Over in the plant cells, you've got chloroplasts, hard at work producing food through photosynthesis. And literally everywhere you look, you'll find ribosomes, these tiny, granular little workers that are responsible for synthesizing proteins.
It's really helpful to map out how all these diverse organelles actually operate simultaneously within the cytoplasm. The nucleus sends down the orders, the rough ER, which is the part studded with those ribosomes, builds the proteins, the smooth ER handles the lipids and the detoxification, and then the Golgi packages the whole shebang up. It is a perfectly synchronized microscopic manufacturing hub.
But hey, every city produces trash, right? So the crucial question is, how does the cell actually clean it up? Well, enter the lysosomes. Now, these guys have a rather morbid, but honestly fascinating, nickname, suicide bags. Basically, they are membrane-bound sacs filled with some incredibly powerful digestive enzymes that are meant to break down waste and foreign materials. But get this, if a cell becomes severely damaged, these lysosomes can actually burst open, releasing those potent enzymes to completely digest their own cell. It's the ultimate cellular self-destruct mechanism, all designed to protect the rest of the organism. Wild, right?
Now, let's talk about something that is truly bizarre. Most normal organelles rely entirely on the nucleus for their genetic instructions. But then, we have the mitochondria. The famous powerhouse of the cell doesn't just sit there and produce ATP energy. It actually has its own circular DNA and its very own 70S ribosomes. It is semi-autonomous. It literally carries its own genetic blueprints, completely independent of City Hall, which allows it to replicate entirely on its own.
Okay, our final stop. Section 6, Osmosis and Diffusion.
It's absolutely crucial that we clarify the distinction between these two forms of passive transport. So diffusion is the spontaneous movement of any gases or liquids from an area of high concentration to an area of low concentration. Think of it like spraying perfume in the corner of a room and eventually smelling it everywhere. Osmosis, however, strictly, and I mean only, applies to the movement of water across a selectively permeable membrane. It's the exact same basic physics, but osmosis is exclusively the water channel.
As you can see illustrated here, the molecules naturally and spontaneously just flow down the concentration gradient. They move from the highly concentrated area over to the lower concentrated area until they reach a perfect, happy state of equilibrium. And because this is passive transport, this entire process requires absolutely zero energy, zero ATP from the cell itself. It just happens.
And if you want some tangible, real-world proof of this, our source material outlines two classic super visual experiments. First, if you take some shriveled-up raisins and drop them into pure water, which is a hypotonic solution, you'll actually watch them swell up as water rushes into the cells via osmosis. Second is the potato oscilloscope. If you carve a hollow cup into a potato, fill that cup with a sugar solution, and then sit the whole thing in a beaker of pure water, you will literally watch the water level rise inside the potato. The water's moving straight across the living membrane of the potato just to balance out that sugar concentration. It's amazing to see in action.
As we finally wrap up our tour of this microscopic metropolis, I want to leave you with a kind of provocative thought. We saw that the nucleus is the undisputed control center, right? But if the mitochondria, the very powerhouses producing the vital ATP energy needed to keep this entire city alive, have their own DNA, their own ribosomes, and replicate entirely on their own, who is actually running the cellular metropolis? Is the nucleus truly the boss? Or is it just hosting a really powerful, totally independent energy company inside its walls? Keep that in mind the next time you think about biology. Thanks so much for joining me for this explainer, and keep exploring.